Plasma membrane, chemical structure, lipids and proteins. Plasma membrane, structure and function. Structure. The plasma membrane, also known as the cell membrane, is a semipermeable membrane that surrounds the cell, providing protection and regulating the movement of substances in and out of the cell. Chemical structure. The plasma membrane is primarily composed of lipids and proteins arranged in a fluid mosaic model. Lipids, 40 to 60% of membrane. The most important lipids in the plasma membrane are phospholipids. Phospholipid bilayer. The structure consists of two layers of phospholipids. Each phospholipid molecule has a hydrophilic, water-attracting, head and hydrophobic, water-repelling, tails. Other lipids include cholesterol, in animal cells, which stabilizes membrane fluidity. Proteins, 30 to 50% of membrane. Integral proteins span the entire membrane. Functions include transporting molecules across the membrane, example channel and carrier proteins. Peripheral proteins, attached to the surface of the membrane and involved in cell signaling and structural support. Glycoproteins and glycolipids, involved in cell recognition and communication. Carbohydrates, attached to lipids, glycolipids, or proteins, glycoproteins. Play a role in cell recognition and interaction with the environment. Functions. Selective permeability, controls the entry and exit of molecules like ions, nutrients, and waste products. Protection, acts as a barrier to protect the internal components of the cell. Communication, contains receptors that allow the cell to receive chemical signals. Transport, facilitates both passive, diffusion, osmosis, and active, energy-dependent, transport, cell signaling. Proteins in the membrane interact with external signals to trigger responses within the cell. Models of plasma membrane. 1. Overton's lipid theory, 1900. Description, based on the permeability of the plasma membrane to fat-soluble substances, suggested that the membrane is made of a lipid layer that allows the passage of nonpolar substances. 2. Langmuir's phospholipid monolayer model, 1917. Description, Langmuir proposed a monolayer of phospholipids with their hydrophilic heads facing water and hydrophobic tails facing away. 3. Gorder and Grendel's bilayer model, 1925. Description, proposed the idea of a phospholipid bilayer, where two layers of phospholipids are oriented with hydrophilic heads outward and hydrophobic tails inward. 4. Danieli and Davson model, 1935. Description, suggested a protein-lipid sandwich model, where the phospholipid bilayer is coated with proteins on both sides. Features, phospholipid bilayer in the center, protein layers on both surfaces. 5. Robertson's unit membrane model, 1959. Description, proposed that all cellular membranes have a similar structure of 3 layers, a lipid bilayer sandwiched between 2 protein layers. 6. Singer and Nicholson's fluid mosaic model, 1972. Description, the currently accepted model of the plasma membrane, phospholipid bilayer with proteins embedded and moving within the lipid matrix. Proteins are integral, spanning the membrane, or peripheral, attached on one side. Summary of major models. Thank you for watching.